Okay, so the attendees they are coming. Let's wait a few seconds that they fill in. And like there's oops. Okay, still some people joining, so let's wait a tiny bit.
I mean, from my side, we can easily wait 5 minutes. I have nothing really scheduled afterwards if you want to.
Okay, okay, okay. But we should be more or less to the number of the registered people. But I welcome you to the 11th FNIP webinar. This is a webinar, an interdisciplinary webinar we organized with the idea of discovering the connection between novel techniques in computation, computational techniques, artificial intelligence, machine learning, neural networks, and how they apply to scientific research, in particular in photonics and neuroscience, but also how, on the other way, they can get some input by the research in photonics and neuroscience to advance and inform different ways forward for the computational techniques. So I just wanted to spend a few words on our initiative. FNIP stands for Focus on Optical Neuroimaging and Photonics. The initiative is based on a group of organizers mainly in the University of Padua in Italy, but also on other countries. And here I am representing the part of Padova, let's say. I'm Francesco Lorenzi from the Department of Information Engineering, and I'm a postdoctoral researcher in photonics. I'm currently working in theory of nonlinear optics and the connection with Bose-Einstein condensates, which is the topic of my PhD in physics of matter, and I'm especially curious about solitary waves, nonlinear waves in general. And I'm here with other two organizers, which are Dr. Matteo Bruzzone.
Yeah, I'm a biologist by education. Now I'm working on the evolution of neurons and neuronal circuits at the University of Fribourg in Switzerland, but I started my PhD and my early postdoc on microscopy, two-photon microscope, precisely volumetric imaging in zebrafish larvae. And that was my point of contact with the initiative. And the last organizer is Marco Brondi.
Ciao, I'm Marco. I am an old school biologist. I'm old because I'm old of age, but the point is that I've been working in neuroscience and molecular biology for the last 15 years. And now I'm in Pisa in the Institute of Biophysics and working on the development of new sensor and reporter for neuronal activity, together with electrophysiology and classical stuff. Yeah, Francesco.
Yes. Thank you. So the group is actually more extended than our three. We are actually organizing periodic webinars every 3 or 4 months, and also we organize a conference in presence every couple of years. The third edition of this conference was in March of this year, so just passed. The fourth one will be in the spring of 2028. So in the meanwhile, we will have quite a number of webinars. And what is this initiative about? As I said, it's about photonics and neurosciences. So the topics of the initiative can be divided these three categories: the let's say optical technologies and photonics and their application, for example, to microscopy of different kinds, and then as an application, but also as a main topic of discussion in our group is the topic of neurosciences, in particular neuroimaging using optical techniques. And these two topics have a strong interaction, as we will soon see, with the techniques of simulation and data analysis that can be applied both to physical platforms of light propagation and interaction with matter, but also to the study of neuronal circuitry and more. So the present this webinar is dedicated to the second topic and to the third topic, but there is a bit also of the first. So actually, these topics are not closed boxes, but they have a lot of interaction, as we will see. And it's about artificial intelligence and the interaction with the topics of our interest. So the idea behind this event is that there is a bidirectional relationship between topics that are pertaining to our initiative and the novel computational techniques. In particular, what is really striking is that not only as we know this new and not so new actually, no? Machine learning techniques can be used in our research, in our practice, and to analyze data, to extract physical laws and laws pertaining to networks, etc., etc. But also there can be the other way around. So starting from the knowledge gained in photonics and neuroscience, it is possible to get some techniques that can be used for the development of machinery, of artificial intelligence and neural networks, and also in general as a inspiration principle for new approaches. Not only that, we can also implement, as we will shortly see from our first speaker, we can implement these computational platforms with photonic technologies. So our first speaker of today is from France, from CNRS, and I will shortly introduce him later. But the topic he will talk about it's the training of physical system implementing neural networks and an analysis of their scaling laws. So the other way around, as I said before. Our second speaker of today will move a bit more to the topic of neuroscience and will show us a striking application of these computational techniques. So let's say we have the two sides of the arrow that orients our exploration of today. And lastly, we have the Professor Dorigo, who will talk with us about a general perspective of the usage of AI in science and the presence, let's say, of AI in science. So I would just want to now tell you how to manage the practicalities of this webinar. So actually, you all have the possibility to send us questions even during the talks. We will keep them for the end of the talk to propose them to the speakers. And you can tap the button of question and answer, and we will receive them. Also, if there are questions that still remains after the talks, of course, we have only 5 minutes of question and answer, so it's not much. We will have a follow-up session afterwards, after let's say 5 p.m. And over there, feel free to raise the hand, and you can also ask an oral question directly. So this is all for the introduction. I'm very eager to get to the presentations, but first I ask the audience if you have some questions on the webinar and on the FNIP initiative in general. Okay, seems not. I noticed that it's quite early, but I asked Professor Brunner if it is okay to anticipate.
Sure. But I mean, for my side, it's all okay. It's up to you.
Okay. Maybe we can have a little bit more of a question and answer then. I think it's beneficial since.
Okay.
Okay. So let me just introduce. Let me just introduce Professor Brunner. So. The Professor Daniel Brunner is a researcher at CNRS. He's with the Femto ST Institute in Besançon in France. His interests include novel computing using quantum or nonlinear substrates with a focus on photonic neural networks. Beyond other prizes, he was awarded the IUP, Institute of Physics 2010 Royce Prize, and the IUP Journal of Physics Photonics, nice journal, and the Emerging Leader 2021 Prize, and the CNRS Bronze Medal in 2022. He's also holder of a ERC Consolidator grant and a pilot of the French PEPR Electronique project of France 2030 initiative. So, Professor Brunel, the floor is yours.
Okay, so thanks for the kind introduction and for the seminar. One word at the beginning, I'm not a professor. I will never be as long as I stay at the CNRS. I think it's the same in Italy with the CNR. But I don't care about titles. But not that someone who takes this serious then comes back is like he's making claims. Yes, so thanks a lot for the introduction. I would almost say that I have stopped my interest and activities in quantum substrates, and I really strongly focus in the last years on the optical things. And with this, let me start to share my slides. I need to request it from you first. Okay, so good. You can see everything?
All
You can see
clear.
my slide?
Yes.
Okay, great. So, yes. Good. So let's start. I'll try to stay on time. I'm a bit of a habit of going over time. So could you like two minutes before the end or 5 minutes before the end of my regular speaking time, just give me some indications?
Sure.
Okay, great. So yeah, as introduced, the title is Model Free Training of Lasers, and then I actually used the title of Clio here, Spiking Photonic Neural Networks. The original title for this seminar was Scaling Laws. I removed the spiking. I put back the scaling. I forgot to update the title. So I apologize for this. If there's some time left at the end, I will also maybe do a quick tour de ville for the spiking work. I am really happy of being in this kind of, neuro perfused environment because actually I did my PhD in quantum optics and then I changed to this whole, unconventional computing business in 2010. And at the beginning, I took a lot of detours through neuroscience conferences to get a bit of background there, get a bit of inspiration that is orthogonal to the usual machine learning fields. So I use these backgrounds a lot for motivation. But first, I want to give a big thank you to especially everybody here on the left side, which is our team here in Besançon. Several people here that are distributed across multiple projects. Some of them are related to optimizing optical systems for computing. Some are, related to using specifically lasers. Some of them are related to the 3D photonic integration work we're doing. And to my main collaborators here from Poland, they are providing the funny looking but great for computing semiconductor lasers that are really now the workhorse in my team to do this kind of physical system computing implementations. Okay, so I think I go usually rather quickly of this motivation slide is actually a couple of years old, but usually these kind of unconventional computing talks start with something like this. On the left side, you start with the neural inspiration that led to the neural network, where the neural network is a very strong simplification of a biological neural network, where we have neurons. Here, they are just some nonlinear unit. They're connected among themselves and to the input information with connections. They might be cascaded in several layers. They might only be a certain, maybe one layer, and then we go directly to the output. Maybe these layers are structured according to, convolutional kernel. But let's say that's the generic principle. And then what you do is you inject information here, you measure the output, and in most cases, what you do is you have some examples here for which you know the output that you expect. And then you wiggle, modify all of these connections until your output is as close as possible to the expected example. And if you've done your job right, then if your network then sees data from the similar kind of class, it will still be able to answer even if it has never seen this particular example before during the training. And from here, we then jump to the digital architecture where we say, all of this stuff does not look at all like this stuff, and therefore it's inefficient. But lately, I'm taking a bit of a step away from this, especially then of the particular quantitative arguments. And I say, why do we always have to focus on this kind of, contrast with digital electronics performance? And we just take a step back, remember that we're doing physics, and try to look back a bit more at nature and why it's more inspiring and also scary good. So there I have usually 2-3 things to say. The first one is the bandwidth angle. So here at the left side, you see a spiking trace of a biological neuron. It's just from Wikipedia, measured with patch clamp technique. And you can see the timescales, 50 milliseconds, give or take. And on the right side, you see the response time of human test subjects to certain tasks. I will not go into detail, but what you can see here is that this, for the simplest of tasks, it crosses the significance threshold at only 250 milliseconds, meaning within, let's say, 5 to 10 of spike times between flashing the information and the human answer to the task. So this includes everything, the signal, the optical signal in this case, hitting your retina, optic nerve going to the back of your brain, to the neocortex, doing some processing here, then bouncing to some other signal processing or processing areas in your neocortex before it then travels around your arm and your finger hits the button to give the answer. So, and what turns out, this is actually at the propagation, signal propagation delay limit of really the signal hitting your retina, bouncing around your brain, going down the arm. So there is no big kind of recycling, overlooping going on in the brain for these kind of very simple tasks. It's just physiologically not possible to reach to these short time scales. Similar arguments exist when we look at energy efficiency. Forget these things. It's from a already, let's say, seminal paper. Of course, there can be an order of magnitude or factor 3 somewhere involved. There's some debate. But essentially, what is clear is that within the energy consumption of the neocortex, 90% is dedicated to spiking and signal propagation, which is what we say or what we assume is basically constituting computing. On top of this, we have all of the learning mechanisms and everything going on with the remaining potential 10% of energy budget. So this is very, very, very efficient and very close to the limit. And now I do a, let's say, back of an envelope, hand-wavy comparison to show you where we are technologically. So this is a very crude argument, and I'm happy if you challenge me and say, but you need to multiply this by a factor of 100. No problem with this. But we have the time scale of a single transistor gate. I equate that to basically a transformation by a neuron. And then we have the inference time of a neural network computer running on a cutting-edge GPU for not too complex models, and that is around, several hundred microseconds or a millisecond. So you can easily see the, 10 to the minus 12, 10 to the minus 3. You can easily see the 9 orders of magnitude difference between in here, where here we are talking about 1 order of magnitude defacing between the individual component timescale, which is the neuron, and the system reply timescale, which is the reaction of the human. Similar arguments, they are less clean in this case, you can do for energy efficiency. Switching a single transistor takes 10 attojoule. Performing the most fundamental transformation in a classical digital computer using a floating point operation is around 1 picojoule. So again, we have like 6 to 9 orders of magnitude, depending on what you use here, of difference between all of these different operations. And this is, I think, where we really need to learn, and we need to focus on doing things in a very different way. The other one, the other feature I want to have a look at is the scaling of these systems. So this here is from a great paper by Kwabena Bohan here. And what we see is that essentially the power consumption of the biological brains grows linearly with the number of neurons they have. So, and the problem is that we now say, okay, the human brain with 86 billion neurons uses around 20 watt and has a scaling exponent of 1. Okay, so now let's assume we have a scaling exponent of 1.5, and then we drop from a 20, if we say we keep the power budget constant, then we come from 86 billion neurons, we drop to 18 million neurons, which is going from a human to a mouse, more or less, by essentially shifting the scaling law from this linear relationships to a 1.5 slope. Or if we assume actually a quadratic dependency with number of neurons and the power consumption, then we are down to 300,000 neurons at the same energy or power consumption budget, which corresponds to a fly. So, and the sad news is that essentially our current digital electronic ecosystem works with that component, with that exponent, with that scaling exponent, at least for fully connected networks, which are really, especially when we talk about large language models, is really the biggest part of transformations going on. So this is these two slides are essentially there to say we need to change something fundamental. We cannot just make some transistors smaller. We cannot just, tweak some bus architecture and suddenly gain 6 orders of magnitude in terms of efficiency, or even scale the scaling behavior of our computing system. So this is more or less the context in which the work that we are doing here in the team are carrying out. And it has several directions. 3D photonic integration is one of them, because you need this 3D in order to make things scalable. In situ hardware learning is another, and this is the main part of today. And just not to use digital electronics to do the nonlinear transformation, the neuron substrate, so to speak. This is the other part of today. Okay, so first let me start with, I leverage these kind of hand-wavy arguments quite a lot. I do like reading maths. I'm maybe not the best person deriving it, but I think most of it is sound, and we can talk about this if you want to. Let's have a look. How can a laser compute? So on the left side, you see the emission of a semiconductor laser when it has many, many modes. It means its diameter is much larger than the wavelength, because if it would just be the wavelength, it would be just a beautiful Gaussian emission, which is great for communication, optical signal communication in telecom networks. But it's boring for computing, because then you only need 3 variables to describe that optical signal in a laser. You need the amplitude of the field, the phase of the field, and possibly the carriers in the semiconductor laser. So it's not a very complex system, and therefore it's also not computationally rich. So if you make this laser larger, you can see you have a lot of speckles, a lot of wavelength features in this entire area. So this is essentially the scaling factor, how many different responses we can implement in this area. So now you see what happens. We have here our semiconductor laser emitting light. We inject information with another laser, and we perturb this information injection by scanning it around the surface. So this is just a proof of concept. Imagine this would be, for example, a pedestrian or the reflection of a pedestrian walking in front of your, autonomous or pretending to be autonomous driving car. And then you want that the laser is like providing a computational response to this. So here there's nothing going on. And this is because the injection as well as the laser, they're not on resonance. If you put them on resonance, the situation changes dramatically. And you can see when we repeat exactly the same input, we scan the same input pattern across the surface, the laser suddenly is very strongly perturbed by this field. And if you have ever seen spike population simulations in brain tissue, the kind of population patterns they form in this laser, they are very, very similar to the population patterns you can see in neuronal tissue. And this is, of course, not making a one-to-one analogy, but you can see that these responses are very high-dimensional and very rich. And the question is now, how can I learn to make these responses ideal? And how can I then connect them to an output such that they solve a task for me? So the concept we are using here is a single hidden layer recurrent neural network. So the neural network concept is on the top. You have your input information, for example, an image. Every dot here would be a pixel of the image. This is connected to our recurrent neural network using input connection. They are represented by a matrix of connection weights omega in. This network does then this kind of nonlinear complex response to this input, and then we connect it to an output, and here we want the reply. So let's say we show here a hand-written number 3. Then the 4th output channel should be the highest because, of course, the numbers start at 0. And for this, we then have to optimize all of these connections involved to link the input to the output such that this is really then given and is the case. In our experiments, we do this the following. Our injection laser is illuminating a display. This display can, in this case, again, show a handwritten number. From this display to our semiconductor laser that I've shown you before, we put another display which is reflective and has, let's say, grayscales, where we can locally change the reflectivity of the signal before we send it inside here, and that corresponds to the input weights. Then our semiconductor laser does all of these highly nonlinear and funny complex transformations before we then again reflect it off one of these reflective grayscale displays, and we detect it. Okay, so there's a one-to-one correspondence. The great thing now is that essentially from here to here, the signal travels at the speed of light with only a small delay here induced by the response time of the semiconductor laser. However, they can react with 20 gigahertz. I will show some early stage results later that we actually already pushed this to 8 gigahertz. So that means the laser really only introduces a delay of, let's say, if it's 8 gigahertz of 125 picoseconds, and this can even be further reduced. The other part is that all of these transformations are implemented in parallel. So if I want to have a 10 times larger neural networks, I just get a 10 times larger laser. In reality, it's not as simple, but what is important here is that the 10 times larger laser only needs 10 times more energy and not 100 times more energy. And it still computes at exactly the same speed. And this is what is absolutely is not attainable in digital electronics, where the bigger the system, the slower it gets. I skipped the motivation about why a semiconductor laser is nonlinear and why this multimode laser is a network. If you have questions, we can shift this to the discussion section. And I now go to the question, how do we implement this topological optimization, or how does our laser learn to identify a number? So here on the left side, it's my favorite illustration almost by now. You can see almost again a conceptual analogy to what we do. We have a trainer which gives feedback to a student or neural network AI, however you want it, and this feedback is very high level. It is okay, put the ball here. That was the way you did it was good or bad, and this is pretty much all you will be told by the teacher. If you now so this means neither the teacher nor the girl here playing tennis is aware of the dynamics going on in her brain or aware of the network topology inside of her brain. If you now go to deep learning and there the error backpropagation mechanism, it's exactly the opposite. You need to know everything. You need to know the activations inside of the neural networks, i.e. the response of every neuron to particular input, and you need to know the value of every connection inside of this network. And that's not all. You also need to know the local gradient of these neurons for the state that has been shown it as a function of the error. So it's a very, very, very high dimensional knowledge that is required, and this is just not attainable if you have a physical system. You cannot measure the thousands of degrees of freedom, and even if you could, it induces an energy penalty that is so huge that you will never be able to catch up. And this is where we said, okay, this is basically never going to be competitive or work. We want to go in this direction. And in the next couple of slides, I will flash a number of different training concepts we have used. I will again describe them heuristically. There will be a QR code. Almost all of them, or all of them, are on the GitHub from one of the postdocs in the team, Anna Scully. And feel free to download them and play around with them and ask me question later or by email if you want to know something specific about them. So this is basically, this has nothing to do with population-based techniques. This is for formatting error of mine from previously. Just go here and there are the learning strategies, and this is the QR code with which you can get access. Okay, so again, a little reminder, what do we need to do in order to show the system a 100 digit and then get at the output a power that says this was digit number 3, or this was digit number 8, or this was a 0. So in one form or another, what we do is we show a vast number of examples for which we know the right results, and then we repeat this process many times, and during each repetition we wiggle, so to speak, meaning we modify, perturb some of these parameters, and we study how does this perturbation impact the accuracy of our computation. So now there is just different methods of doing this, but they all do this in the same kind of way. You have what we call in the field a forward pass. We send information forward. And the cool thing or the good thing is, if you do this physical implemented neural network computing, the forward path is what is efficient. The forward path, it is what is done with different scaling laws. If you now want to make a model of this forward path in a digital computer that you could then inside of this model calculate all of these gradients as you would in an error backpropagation scheme, then this model that you need to train your system does not scale nicely and linear anymore. It is scaling exactly to these bad behaviors that I told you before. So by using these kind of perturbation techniques, we make use of the efficiency and of the elegance of our physical computer, not only when we use it for compute, but also for the training. And of course, the price we pay is that we therefore need to repeat a lot of times this input information, and we need to wiggle the system and perturb the system in many different directions, because we will never get as much accurate information as you would have in this full, measured system. But because our system scales differently when we go forward, and because it's much more efficient, ultimately we believe, and we are soon to be publishing an article that actually proves this or demonstrates this, that because we use these advantageous features in the forward parts too, this inaccuracy in obtaining gradients is actually more than compensated for by the elegance of the computing algorithm. Okay, long story short, we show the system many examples for which we keep this parameter constant, the weights, then we wiggle these weights, perturb these weights, and repeat showing all of these examples. And we do this in different ways. And then we update the weights on our spatial light modulator for next generation. So the first way what we do in this is when we implemented weights that are not analog numbers. They are binary or trinary, meaning they're either 0 or 1, or they're -1, 0, and 1. The problem is there, even these kind of, repeated data showing mechanisms and wiggling mechanisms, in many ways, they use differentiability. And that is the differentiability of the change onto the loss function or onto the performance accuracy. If you have zeros or ones, then this does not work because it's not differentiable, the changes you can introduce in your perturbation. So this means many algorithms are directly excluded. And here we went back and looked into thermal annealing strategies. And thermal annealing just means we invert, for example, if we say the pixels of these spatial light modulators that represent the weights, we take a number of these pixels and we invert their state. Meaning if some of them were zeros, now they will be ones. If they were ones, some of them will be zeros. And with this, we just send then again a number of examples through the system, and we get a performance. Now we basically invert some of these weights, repeat the measurement, and we see if this performance improved. Then we keep this modification. If it got worse, we reject this and we try a different modification. So we have optimized and actually developed a complete new form of this algorithm where we just have one single parameter, and basically the quintessence of all of this is the number of parameters that we invert in this annealing concept is linearly proportional to the accuracy of computation, meaning at the beginning you invert many, and the better and better you get, the less you invert. And we make this simply proportional to the accuracy. And this is what is called also thermodaptive simulated annealing, but there you have much more of these parameters to optimize. So long story short, this is from basically showing digital simulations. Here you see now the performance really when we show our laser on a display, a handwritten number, and task the laser to predict which number was this. So, and here are several pieces of information. The first one is the digital linear, and that would mean just I take the number, let's say that's 784 pixels. I have 10 outputs because we have 10 digits. And then I create the fully connected matrix between them. And if I now find the optimum configuration for this matrix, then we get the green bar performance. And this is our limit, because I would say if we do not go beyond this, the computer is not really helping. It's a linear computing. And therefore, this is the benchmark which we need to overcome to really claim our system is a useful computer. So the physical equivalent to this would be the VCSEL off, the laser off, because this makes our optical system linear. And then we train our weights in hardware, as I said before. And then you see that we do approach this green limit, but we stay away from it for quite a margin in many situations. And what this illustrates is how difficult it is actually to simulate and to train a real system, and not just to write a simulation in MATLAB or in whatever programming language you use. And then we switch the laser on and we compare the performance where we use Boolean weights, i.e. zeros and ones, or trinary weights, and you see that there's a significant benefit when you actually allow for the -1s and not only ones and zeros. But in all cases, for all handwritten digits, we are not able to surpass the green linear class. So now we switch to analog weights, in our case 8 bits, 9 bits, 10 bits, doesn't really matter. And what this allows us to do is we do population-based sampling. So the simplest illustration you can see here on the left, what is population-based sampling? It means for my current population, I have a certain configuration of these weights. They usually vary to some kind of statistical distribution. And this is what you see here. The green is the center of the statistical distribution, and the blue are the examples that are randomly selected. Each example is now one of these configurations of my weights. And for each of these configurations, I send, let's say, 1,000 examples of handwritten digits through it and calculate the error at the end. And now what I do is I then compute for each of these statistical distributions a performance, and then depending on the algorithm I use, I calculate a next generation. So the very simplest of things to do would be just what you see here. I initialize my system randomly, and then I create a population around it, to a certain sigma, and I measure the performance, which is here color-coded. Yellow is great, red is bad. And for this entire population, I look what was the best candidate. I make that candidate the center of my next distribution, and then I distribute it around it again. So that works. It buys you constantly until the good configuration of your system, but it's very inefficient. So there's other algorithms such as CMA and PPG. What they do is they adjust the width of this distribution such that the distribution that the direction that points to my optimum direction is very wide, because there I want to aggressively sample, and the directions that are orthogonal to the direction towards the minimum, they're sampled very little, because I cannot expect to find big improvement there. And they just use different kind of techniques for this. Genetic algorithms, they're doing something very similar. Particle swarm, they're doing something very similar. They just always have these populations, and then they create an update. Where particle swarm is different, but yeah, all of these other kind of algorithms. So another technique is gradient-based and gradient approximation techniques. So these could be called finite difference. So you could imagine, instead of making a population, I make a random initialization of my connections, and then one connections I change by epsilon. I calculate how much did I get better or worse, and then I do a gradient descent of this one parameter, and then I just randomly cycle this around. And it turns out that this is very inefficient. It works, but it takes very long, and there's a stochastic method called SPSA, where you perturb all weights at the same time by an epsilon in a random direction. You measure the performance, and then you perturb the weights again, but now in the opposite direction by epsilon, and you measure the performance again. So instead of now having a large population for each update, you only need to measure twice. And this is the big advantage, but you will see that basically they are attractive for not high complexity systems, but not for the real big systems. So the first thing I want to show you here is what happens when we train only the output weights of our laser neural network, and we scan also the size of our neural network. All of this can be found in this arXiv article. It will not be published anywhere else. Please cite the arXiv if you find it interesting. There's also much more calibration data and everything on this kind of experiment. So it's a very interesting article to read. And you can see here that with our one laser, we can realize around 2 to 2,500 neurons. This laser needs like 50 milliwatt of pump power, has the potential to react at 10, 20 gigahertz bandwidth. And therefore you can see where we get very energy efficient. But again, sadly, we are not crossing the linear threshold. So what we do now is, at the output dimensionality, we fix the 2,500, and we now also include the input weights, the first SLM in this optimization. And then we do the same trick again, and you see that if we increase this input dimensionality, you can see that we have a very strong improvement of performance. And for this, the digit number 8, we then, for the first time, really in an analog hardware that is having no pre-processing or post-processing in digital electronics, we surpass this linear threshold. We have a much better laser than this one, and the results of this one are shown here. So here we have roughly 93% across all the digits, whereas our best laser was at 96.7%. Again, without any no post-processing for all the digits in the handwritten digit classification task. The problem is that just this laser died and we could not repeat it and make this kind of size-dependent scan. This is why I show you the different one. And that laser actually had the correspondence of 5,600 neurons inside of it. So again here, same game. I show you the linear threshold. Then what happens with the VXL off, now optimizing it in hardware, but now with these analog weights. And then we have here two comparisons, this kind of gradient approximation techniques, which is the SPSA, and this kind of population technique. And you see that the gradient approximation technique again fails in most cases to overcome this linear threshold. But if we switch to these population techniques, we overcome this and sometimes by quite a margin. And this is the first story or the first punchline here. It is really the pairing of the system that you use for computing and the algorithm that you need to consider. You cannot basically treat them individually. Different systems have different kind of preferred algorithms. And this is something where a lot of work has to be done in the near future. Okay, so this is a quick outlook now to the part I forgot to add to my title, which is the scaling laws. And here, what you
Sorry,
see.
you have 5 minutes. Sorry,
Okay, yeah,
Yeah.
I think I have only 3 minutes, no?
but say 5.
Okay, 5, good. So what you see here is essentially the graph that caused the current hype in large language models, ChatGPT, however they are called. And what this shows is that there's a power law scaling behind the accuracy of these neural network models and the energy you spend to train them, as well as the efficiency when you increase the size of the neural network. So it basically says if you have a very small neural network with only 100,000 parameters, actually in my experiments or in our experiments, we're approaching this number soon, then you get this kind of scaling law. And you can see that the performance still reduces. So if you would have infinite amount of energy, you could still train the system for 3,000 years and it will arrive down here. But the big benefit of making the models bigger is that essentially you converge quicker before this tailing off happens. And this is what we call a knee in the performance curve. And if you track this knee, you see this maximum efficiency learning dependency. And this meant here for these models with 1 million weights around here, they never succeeded in training at that time. It was from 2020. Never succeeded fully training the big model. But you could see that for the large models with millions of parameters, you got to big performance. And this showed to investors, the field, everybody, okay, what we have to do right now is we have to aggressively increase the number of parameters from millions to billions and trillions. Yeah, this is billions, obviously not millions. And then we will land down here, and potentially down here is where the interesting stuff will happen. So, and we are trying to arrive at something very similar in my team in the last years. And just the last year in particular, we made great progress in this direction. And for one particular task, I want to show you this. So here you see the mean square error for handwritten digit recognition in our fully hardware trained system. And here we have developed a certain metric, PCA dimensionality. We already have a paper, 2 papers on this. One, Anna Scully is the first author on the semiconductor lasers. The second one is Mathilde Ari is the first author applying these metrics to nonlinear fiber propagation. And what you see here is we analyze our lasers with this kind of computational dimensionality metric. And we have a look at very different laser shapes. So they have very different physics and very different transformations inherent to them. But what we see is now we are able to carve out this kind of power law scaling just as we see it for the large language models. So I cannot say much more because this is still under preparation, this manuscript. And since now we have, by now we have developed this technique much further, and we get very, very great results, where we can actually see that now for different systems, semiconductor lasers, nonlinear fibers, they all fall on the same scaling law that uses our computational dimensionality metric, which is not the PCA dimensionality, and the mean square error or cross entropy loss. And we essentially are now able to find the scaling laws of natural computing in our physically trained hardware. And finally, what is even better and cooler about this is for one of the systems, the nonlinear fiber propagation, we also have a numerical model. And everyone who does this knows that it's very difficult to exactly match a numerical model to the physical system. And also in our case, it is not the same. However, if we, I mean, the postdoc behind this work, or the postdocs behind this work, they spend a lot of effort in making the agreement good, but it's not perfect. So if you scan the system parameters, this agreement could ideally be better. But as soon as you project it on these computational scaling metrics, the agreement between numerical model and experiment, as well as overlap with data coming from a completely different experiment, is absolutely perfect. It is one line. And essentially, this is a big drive in my team in the recent past. How do we get to these metrics, and ideally, how can we then link them to system properties, high-level properties like size, etc., design properties. Okay, so with this, I come to an end. Here, just a very quick snapshot of the 3D printing that we do. You can see this is basically an axiomatic tree inspired 3D photonic integration structure. And we are now able to identify these digits at timescales that are much faster. This is very preliminary, but this is essentially showing you, let's say, it's a promise for the future with this kind of system. And with another semiconductor laser system, you can see here, actually I cropped that x-axis. You can see what we do here is signal reconditioning, where black is the ideal signal, gray is the starting signal before optimization, and orange is what we get out. And you can see that I think in this time trace, if you just put a threshold at 0, our analog neural network does absolutely no mistake in this digital sequence reconstruction. And the importance here is that the x-axis scale is nanoseconds. So this is full real time and now approaching really the bandwidth of the semiconductor laser to provide this entire computing result. I cannot say much more also about that system because we are still actively doing experiments and writing, well, hopefully soon writing the paper. Okay, and with that, some advertisement. We have winter, summer. I mean, April is not really winter or summer, but Les Ouches in the French Alps in Chamonix. It's a beautiful place that you can see here, where we have great speakers on machine learning and photonics, in photonic design, and the whole interaction between these kind of directions. So if you're interested, have a look at that, and I would be delighted to see you there. And with this, I skip the summary slide, and I thank you for your attention.
Thank you very much for the very nice talk. Now it's time for some questions. We prioritize the one from the audience. Let's see. So while the audience think of some questions, I actually have a curiosity. You mentioned towards the end of your talk the another platform with respect to the vertical cavity laser, and you mentioned the nonlinear propagation in an optical fiber. I got curious about it time ago because I came to take a look at an article by Giulio Marcucci and Claudio Conti, and
Yes.
they are using like a single mode fiber, and they are using the nonlinear Schrödinger equation
Exactly
as
the same. Yeah.
is the same. But it's very nice. How would you compare these two platforms for the targets that you have? Like also on the perspective of the future developments. It's a big
Oh, now
question.
you're trying. Now you're trying to tell. When I have two kids, you want me to tell which one I
Yes,
prefer.
exactly.
So I mean, first the fundamental difference is the lasers I've shown you. There, this nonlinear expansion happens in space, in the spatial domain, whereas in the nonlinear Schrödinger equation propagation, you go from the spectral domain. So you have a femtosecond, input pulse that you modulate the spectral phase, and that then broadens spectrally as it propagates through the nonlinear fiber according to the nonlinear Schrödinger equation. So that's the first difference. We find that right now, the semiconductor lasers are much easier to scale up, but they have a certain limit and they're much more energy efficient in what they do. Ultimately, because the nonlinearity in the semiconductor laser is an electro-optical one, you modify the carrier concentration in the quantum well, and that talks back on the optical field. The nonlinearity in the nonlinear Schrödinger equation is, carrier effect predominantly. And that means it needs much higher energies, optical energies, but at the same time, it gives you terahertz bandwidth because you don't need to move electrons or carriers. So I think to replace the current generation NVIDIA processor, none of our systems is actually good. That's not the purpose. It is really to do this kind of analog closed loop computing, learning the physics, etc. But I have very high hopes for an entire new class of physics experiments where you need to already process high bandwidth data, project it down, for example, to a latent space to save storage, etc. And there, your physical computer needs to match the timescales of your experiment. So that means the semiconductor experiments are great for anything that's slower than nanoseconds, or let's say 100 picoseconds. Whereas with the femtosecond laser and the nonlinear Schrödinger equation, you could actually do really advanced experiments and metrology concepts, etc. Really at few cycle bandwidths because you modulate your input. So this is the kind of difference I would say in between. So it's true we can make the laser much bigger. The semiconductor laser is much more energy efficient. It's an easier setup, but it has temporal limitations. So if you want to really dive into this ultra fast dynamics and do some computational ink the loop experiments, then you need to shift to the other system.
I see. Thank you for the answer. Very interesting. Some other questions?
Good.
It's
If not,
super cool. There's all these chips that we can get. Like, so if I wanted to try this out, I know I could get a TPU. I know I could get an NPU. What's the thing that's closest for us to be able to try it? Is there some way that we can try our systems on it?
on it, what is it? The laser?
It being, yeah, one of your systems. That's what the 3D printed thing is supposed to start to solve? Is that?
So the 3D printed in the end is supposed to really mimic this kind of synergy between 2D neuronal substrate of, the neocortex. Basically, all the neurons are on the surface more or less, and the medium long range connections go through the volume. And this is ultimately why all of these connections fit inside of our skull. And when we look at the electronics, everything is projected onto a 2D substrate. And this means nature is scalable because it uses this synergy between 2D and 3D. 2D for the computational substrate, 3D for the integrational substrate. And the 3D integration work we are doing is exactly following this inspiration. But this is really, let's say, we are fastly improving, but we are nowhere near this kind of programmability scale. This is really a long-term research. Regarding for example, doing neural computing, we have done other systems. They're all proof of concept, but they're hardware based. We created a spiking neural network with 40,000 neurons that we also trained. And there we used the latency encoding to enforce strong sparsity. And we show that basically until a sparsity of almost reaching 90%, we have almost no loss in computing performance. And even if we go to 98% 98 or 98, 99.8% sparsity, meaning only 0.2 of the percent of the neurons spike, we still get close to 40-50% accuracy. So of course, if you want to have a high reliability computer, then this is nonsense. But if you want to have a very fastly reacting robot or very efficient robot, then this is great because, you know, so it's like, and the interesting part here, the mechanism we implemented needs one variable tuning, and you can switch between using maximum accuracy population sparsity to maximum efficiency. So it's a very elegant switching mechanism. And this system here is, we could make this much, much larger, but it's very slow. So any of the systems that I have shown, they are not really commercial. They are not really easy to handle for anyone from the other fields and disciplines. Getting there, this is what we are trying to do now, and we are hoping soon we provide like a web interface at some point where you can upload your data, play around with all these algorithms and systems. Trying to build one yourself, I don't recommend. They can be quite
Definitely don't want to build myself, but yeah, I'd love to play with it if you guys have it.
So if you want to play around with this, if you have some data that interests you, contact us. And if at some point we have this web interface going, I will let you know.
Good. So I would go to the second speaker. I will keep the other questions for the follow-up session if Daniel Brunari joins us. So thanks Daniel again, and even if we are a webinar, thanks again. So
thanks.
So I let the stage to Matteo.
Okay. Now it's time for the second speaker of today. He is Professor William Buchser. He's an associate professor at the Washington University in St. Louis, and this group combines automated liquid handling, nanoliter dispensing, high content confocal imaging, and machine learning to study how single neurons of different subtypes become vulnerable to neurodegeneration. So, Professor Buchser, the stage is yours.
Thank you, Matteo. Hey guys, hello from across the ocean. Really cool to hear Daniel's talk, and I love the intro he gave to get everybody just thinking about the nervous system and the complexity and just, you know, the truth to that, whatever it was, 10 to the 6th, 10 to the 9th difference between the truly amazing electronic neural networks that we have now that we use for everything, but still how much more amazing our brains are. And so we're definitely coming from the aspect of when real biological brains break, aka neurodegenerative disease, how can we fix that? And I'll get you thinking about that in just a second. But at the same time, it's super confusing to everybody who we talk to because we are, just like the rest of you, neuroscientists, but also using all these neural networks and all these artificial systems to help us in a lot of different ways. And so, yeah, we're gonna kind of look at the whole brain here, zoom out. We are going to do some single cell stuff, but not in terms of how the neurons are behaving, because we're really interested in why they are degenerating in certain situations. And so we will start with just thinking about neurodegenerative disease. Oh, and by the way, anybody, including the panelists, feel free to let's just turn this into a discussion at any moment. You feel free to interrupt me and don't mind at all. So you guys can hear me okay, right?
Yeah.
Okay, thanks. Neurodegeneration, obviously, it's pretty straightforward. Anything usually happens to adults, older adults, but it can happen to kids. We see people in the neuro clinic who are 20 years old and they have neuropathy. They can't use their cell phone, right? Because even though they can still move their muscles, losing the ability to feel makes it you can't operate tools. It's really hard to use stuff like a cell phone. So it's super debilitating and one thing if you're old, but another thing if you're 20 years old. Those are more rare, but they're usually genetic. And so the table I have on the left is just getting you to think about, there's a lot of people that are affected by this, whether it's Alzheimer's, glaucoma, macular degeneration, Huntington's, Parkinson's. Most of these that affect the most people are not just one simple genetic change, right? It's polygenic. There's many different variants, many different mutations that lead to the disease. It would be nice if it was monogenic because we could target one specific thing, but all the different variants and all the different genes is what makes it so tricky. So it's often thought of as like an idiopathic disease, right? Because it's hard to track down the genetics. But we still want to do that. We still want to say, hey, if somebody comes to us, especially if they're younger, with one of these neurodegenerative diseases, can we figure out what the mutation is? Because we're only, well, it's happening right now, right? But it's possible that within a year or two, we could make a custom gene therapy to edit that specific mutation. And so the hard thing is not as much fixing the mutation, it's figuring out which mutation is causal. A common example might be the gene MFN2, which helps mitochondria do their thing inside of a neuron. And if you have just a small number of mutations in that gene, you get Charcot-Marie tooth disease, you get terrible neuropathies, both sensory and motor. People usually don't live much past their teens.
Sorry, Professor?
Yes.
I don't know if you share the slides, but we don't see anything.
Well, of course, you just want to look at me the whole time. Dun, dun, dun. There it is. How's that?
Okay. In fact, okay.
Thank you, Mateo. Yeah, I should have said, do you hear me and can you see me? So here, I'll just back up so you guys can see that was the overview. So sorry you missed that much, but that's all good. Okay, there's CMT2A, and then just thinking about what does it actually mean to have neurodegeneration, and this is what it means. So there's intact axons that are all interconnected that Daniel was talking to you guys about. They need to be able to connect to another neuron for them to have any use at all. And in neurodegenerative disease, it's not that the neurons die, they eventually will, but what happens sooner is this, that their axon, that incredibly long wire, even in the brain where connections are relatively short, it might only be an inch between cells, it's still the equivalent of having to drive, hundreds of kilometers. If the cell body is the size of a car, then the axon would be like, 100 kilometers long, even a relatively short neuron. So these are vast, vast distances. And if there's any spot along them that breaks or is degenerated, now you lose connection and you can't send that signal. So this is, when you hear neurodegeneration, this is what you should think of at the cell level. But of course, what are all these genetic mutations? What are they causing to end out allowing the neuron to fall apart like this? And I'm saying fall apart, but in reality, it's actually active. It turns out there's a whole process that actively destroys axons that can get triggered for the wrong reason. And we really want to prevent that. We actually understand that pathway, but we don't understand all the triggers. And so just another kind of interesting note about how we study this is that if you just look at the soma, the easiest place microscopically to look, it tends to not have a phenotype. So the interesting biology happens distally or away from the cell body, whereas the proximal portion of the neuron tends to be healthy for much, much longer. And all the interesting stuff happens distally. And that's what I'm showing you here in this image of this happens to be a rat nerve. And so at more, closer to where all these neurons' cell bodies are, you see lots of intact axons, which are these little circles. Those are the myelin sheaths. And as you look further away, you still see some myelin sheaths, but you see many axons that are just fully degenerated. And Jason and Colin are my partners in crime here for doing a lot of this work. So it's just to get you thinking about neurodegenerative disease. That's why we care. We want to be able to track how mutations are affecting and causing neurodegenerative disease. Okay, but let's zoom out. So this is all taking place in a real brain. This is a picture of a mouse brain. And just like, there's so many tricks and so many different ways to think about this. This tissue itself is just an enormous, amazing, rich data set, even before we get into spatial omics, which I'm going to talk about in a second. And so this is, a histology slice of brain. It's only stained to show nuclei. But yeah, just go with me for a second. So as we look at this level, obviously you can see different atlas regions like the hippocampus, the dentate gyrus, you can see the isocortex, you can see some of the thalamus. But this image, of course, is taken at a very high resolution. So we can go way down and actually see each individual nuclei here. Here you can see nuclei along the dentate gyrus. You can see nuclei of cells that are in other parts of the hippocampus. And you can see that each individual cell, even just with this one dye, has different intensity, different size, has different like heterochromatin and euchromatin inside those little speckles. And this is all real. This is just a real image, right? And so to be able to reconstruct this and really fully understand it in a digital sense, right, where we like, here's every single cell, here is all the cellular features. In this case, we only have nuclear features. Here's all the different cell types we think might be there just by clustering those nuclear, and I'm only talking about what we can see, so image-based features. How do the cells cluster together? Can we find lamina and stria? Can we mark all these anatomical regions, right? So that from the scale of organelles all the way up to the scale of the whole brain, this image is just an incredible data set. And the concept of like data existing at multiple scales is an understatement, but you guys can see that, right? Like if I only knew like, hey, there's 1 million cells here and then they have these cellular features, I'd be missing out on like, how are cells next to each other? Are they part of these contours or little clusters? And then of course, what brain regions are they part of? And so even without adding any other data, the image is really where we want to pull a lot of information. And this is where us and other genomics folks that I work with all the time have many arguments. Okay, so let's talk about that. So you guys might know about spatial transcriptomics, spatial proteomics, spatial metabolomics. There's a lot of now genetic techniques or genomic techniques that instead of just saying, hey, we can throw a brain in a blender and say, here's all the genes it expresses. That was what we did 20 years ago. And then 15 years ago, we said, hey, we can do single cell technology where we can separate individual cells and measure their transcripts or ATAC, which things are turned on or turned off. We can look at single cell proteomics. We still don't know what those cells are. We just know that cell X has this transcriptome, cell Y has this transcriptome. The newest, most fun thing is these spatial omics. Not terribly new, of course, it's been around for 5 years now. Many people are using it. And it's to say, why throw out the spatial relationship between those? And let's keep all of that intact. And so just so that we're all on the same page, this is just a quick didactic thing of understanding the different platforms. So the two most common that you might see in papers or you might use it at cores or different places in your labs. So MERFISH, which is commercialized as 10x Xenium, it's not exactly the same, but they're close. These are the most high resolution, so you can see where a transcript is, precisely depending on how good your microscope is. There's a little dot, that's transcript that was detected by a certain probe. And so MERFISH and 10x Xenium are great because you have like subcellular resolution. The problem with these is that what you lose is, since they're probe-based, you're limited to a certain number of, it doesn't have to be transcripts, but as an example, transcripts that you can ask about, that you can query. So the initial system had about 500, the new system has about 2,000. So you can ask questions about a few thousand transcripts, but you have really, really good subcellular resolution. 10x Visium or SlideSeq is kind of the opposite. So you sacrifice resolution, you're certainly not subcellular. If anything, you're like just about cellular, but not quite, because it's all kind of spot-based. And this is much more like a microarray where you have a patterned substrate that has many, many, many barcodes on it for basically, so that you can, usually 3', you can attach stuff onto there. You can attach transcripts often, but it doesn't have to be. And so this is great because then you just sequence that. And so although you lose spatial resolution, right, each little spot has a spatial barcode that tells it where it was from, you can now sequence the whole transcriptome. So you have whole transcriptome, but you lose the spatial, you have to give something up in the process. Where I am at WashU, we have both of these, and we kind of use both of them all the time. There's also many ways to do this with proteins. So we use something called Akoya Codex, but there's other systems and many other ways to do this that are also fantastic. And this is the simplest version you can think of as like an antibody panel where you can flow through many different antibodies. So the panels we use have about 30 to 50 antibodies and can stain and ask about the proteomic landscape of a tissue. And so again, you're only limited by the resolution of your microscope that you can read this out with. So very easy to get subcellular, but you're not doing whole proteome. In this same category, we also have systems now that can do actual mass spec, spatial mass spec, and those are much more expensive, much slower, but you can really ask questions about much more molecular things than just a set of proteins. And just for completeness, we should say that 10x now has a new system called Atera that there's a limited release of that kind of is the best of both worlds between Xenium and Visium. And I mentioned the MS Spatial there. So the problem for me as a image first neuroscientist, like I think many of you are, is that the molecular folks, I'm doing an us versus them thing. There's not really an us versus them thing, but just is they have been used to doing single cell and identifying cells, identifying neurons, for example, based on their transcriptomic profile. And that's awesome. But it's not the same thing that an anatomist or a pathologist would say, right? An anatomist or pathologist would say, no, this region of the brain is X, this cell type looks like Y, and that's the ground truth. Whereas somebody from a molecular background is going to say, oh, no, it expressed this set of genes, so that's the ground truth. And so there's a bit of argument back and forth, and it's easy for the molecular folks to win there because they're like, oh, we have these 20 markers, but then we remind them, well, you got those 20 markers from the anatomist and the pathologist telling you like that region was expressed in X or Y. And so that's something that we're always kind of trying to deal with. And the answer is that like, if you can use them both, that's wonderful, but you have to be very careful. And that's what I have on the bottom right here, which is the idea of, I call anything that's image first. So, if I wanted an anatomist to look at a brain and say, that's the edge of a certain subnuclei in the thalamus, that's kind of a top-down approach, right? We see the whole brain. We can, we're visualizing it and we're using, in this case, kind of classical knowledge to define that region. Whereas bottom up would be like, we've got the molecular information and we're using the molecular information to define that region from a bottom up way. The only problem with using both, saying like, hey, we think this region, it looks like it's X, right? Perhaps a certain layer of the cortex, like that's how it's defined is anatomically, but we'd love to use some transcriptomic markers to more better define where its edges are. You run into a problem called double dipping, right? Where if you use the marker to help you define the brain region, now you can't use that marker in any kind of other analysis. For example, if we want to say between an Alzheimer's mouse and a normal mouse, we want to measure the transcriptional profile of these certain brain regions. Well, then we can't use those same transcripts to define the brain regions. Otherwise, we're double dipping and we're cheating. So anyway, just throwing that out there. So I really like image first approaches because then we can leverage all of the molecular data and use those in our analyses rather than having to like exclude them because we've used them to define the regions, if that makes sense. Okay, so we're talking about looking, not usually with our own eyes, but usually with computer eyes, looking at all these different scales, right? So from subcellular to atlas level scales where we want to define brain regions. And so that sounds like a classic problem, right? It's the segmentation problem. And why is this interesting? Like, why are we even talking about it? Is it a solved problem? Is it not a solved problem? There's certainly hundreds, if not thousands of tools out there. And the paper we just published and what a lot of what we work on is saying like, yeah, where is that hard? Where does it fall apart? And it's usually the most interesting places. So, in brain tissue, there's some regions that are just super high density. So any classic method, even new methods like cell pose, really have a hard time to distinguish those really high density areas, and that's where the interesting biology is happening. There's also crazy heterogeneity. A lot of these tools are developed off of like, here's a section of tissue that's relatively homogeneous. Even cancer tissue, yeah, there's areas that are less homogeneous, but in the brain, the variation can be incredible. And this is the case in the nerve as well. And then the most fun thing for us is continuity, that like, a single neuron might have an axon that stretches through multiple layers and branches and fasciculates and comes back. This bottom one is the most challenging without having EM level stuff, which is a whole nother nightmare. That's not us, but there's people that do that, and it's amazing, but it's crazy slow, and you can't ask the kind of scale questions that we're asking, which, by the way, is we make perturbations. Daniel was talking about perturbations being awesome, and we agree. We don't perturb the laser, we're perturbing the genome here by using CRISPR to make hundreds or thousands of edits, and then asking about how the phenotype changes. So EM is not really compatible with that. Okay, and again, stop me because I'm gonna go all over the place here. So how do we do this? What's kind of our pipeline? We call it CDOLA, which just stands for simulate, don't label. And you may not have guessed, but just like other ways to think about it, if we want any neural network, whether it's a TPU system, a GPU system, like I'd love to try it with these new optical systems that we just heard about. If we wanted to be really good at all these scales, all these levels of being able to find structures really, really accurately, so we can reconstruct that and understand what's going on in certain mutations, then we're going to need vast training data. And vast training data kind of exists. So there was an effort to do 30,000 brains. It took 15 years, and right, if you want to change anything, you got to go back and have humans go and change it. So it's really, and 30,000, as you guys know, is not enough to make really, really good algorithms, right? You need 30 million, you need 30 billion to make a training set that's just amazing. And so the way that we get around that, the way that we do that is with CDOLA, simulate, don't label. So we build simulations. And a really important note just before, I'll go to this slide, is this is synthetic data, but what's different about other synthetic data that you've heard about is this is not sampled from, we are sampling from distributions, but we're not saying, oh yeah, here's a few images, let's use that to make a GAN and generate more images. That is absolutely not what we're doing, because that leads to what's called generative collapse, where you generate and train, and there's a lot of issues around that. I think there's some that are working better now, but this is completely different. So this is a biophysics-based approach and let me show you. So here we've got at the level of a single cell. So I showed you real nuclei a second ago. These are simulated nuclei. They are just, hey, there's this spheroid. It's filled with heterochromatin and euchromatin that's represented inside of this 3D model. There's an optics engine that's going to help us render this. And those cells have different sizes and shapes and textures and all the things that we measure in a normal cell. But then we can say, rather than giving it the normal Gaussian distribution that we see in normal tissue, we're going to give it a uniform distribution, and we're going to expand the edges of that uniform distribution, because we know that what we've seen in the few, even if we've looked at thousands, we're still seeing only a subset of what might really be out there. And so we can then sample and simulate way past reality so that we can catch all these kind of variations, and that the model's not surprised by it. So we are sampling from distributions that come from reality, but we're rebuilding that all from scratch. We're not generating it with a generative model at all. This is completely directed. So we're directing everything, and we're sampling it randomly. So that's the idea here, right? So then there's a region node. So we take all these individual cells and we say, hey, let's make them into stria. Let's make them into clusters, right? Let's project them into patterns. And again, those patterns can be more extreme than we see in real life. And then we have references like the Allen Brain Atlas. We have references like Spinal Cord Atlas. We have references from the kind of ways that neurons fasciculate together in nerves, even though there's not like an atlas for that per se. And so we can build all these regions into these atlas regions and reconstruct those, right? And then as you can see here, we can, part of the simulation is real things, right? Sometimes the sample gets put on and it gets ripped a little bit. Sometimes like half of the hemisection falls off. All those things are built in, those distortions, the skews. So then all of that gets rendered out with an optics transform. It's not as fancy as what you guys do, basically point spread functions. And then we can switch the rendering either so that we get a microscope-like image or that we get the ground truth. And sometimes the ground truth is just the coordinates of where things are. Sometimes it's an actual image. So we can do various kind of autoencoders, like, there's many different things. We save the metadata, and then whether it's, a UNet, a V, we actually like vMamba a lot these days for these things. And if you're curious, here's what these simulations kind of actually look like. It's a very graphical thing that it's just a little bit easier to look at. Okay, so that's the key. So we simulate a bunch of stuff. Here's an example in spinal cord. So you can see here's the biological examples of, this is kind of the zoomed into individual nuclei. Here's the zoomed out. So you can see finding all those nuclei, finding the regions of the spinal cord. And this is just showing you like, here's this kind of digital twin, real representation of every individual cell and just a few of the things that we understand about it. Here's what this looks like in brain. So on the right would be how the molecular folks see it, right? They do spatial omics. They cluster UMAP based on the transcripts, and they map that onto the spatial thing. They go from the bottom, right, the molecular, back up to the top. And yeah, you can see like lots of cool regions come out of this. But here's how an anatomist would see it. And in fact, all these regions were automatically labeled with CDOLA, just like an anatomist would see those different regions in there. So none of them, I think, is right or wrong. They're just different, right? We're bringing image-based information into the system. The version 2 of this, I know that I'm already close to time, but this is perfect. Actually, this is a great place for us to leave off and have more discussion. So some of what's above is published, and there's there's an online portal you guys can bring your own brain sections to, and you can kind of play with it. A small subset of the simulations are available on Hugging Face. We're going to put more up soon. The new version is because of a consortium that we have at WashU, which is called, very funnily, WooSmack. So the SMAC is the small mammal core, where they're putting a lot of neurodevelopmental and neurodegeneration associated genes into mice. And then we have massive both molecular and image-based phenotyping going on. And right now, when they do the brain slices, it takes a team of 4 people an entire week to come up with all of the labels of just like 8 different brain regions. And part of the reason it takes so long is that they're really careful about the boundaries between where one region starts and another stops. And that's all led by a neuroanatomist, but it's just incredibly slow because they're giant slices. We have now a version of this for them. We had to just rethink it a little bit because, of course, one thing we didn't consider in V1 is that in reality, when you slice through these brains, you get slightly different angles and slightly different, these are all coronal sections. And so our newest simulations actually start with a 3D brain model. And then we slice randomly through it. Then we can extract the regions from that slice and add neurons and add the simulations on top of that. And so the data set got like thousands of times bigger because now, rather than just having always one perfect coronal section that we mess with, we're now slicing at every possible angle at every region of the brain. And so the data set is just massive. We're trying to work with NVIDIA to host it for us so that it's a little bit more reasonable. But so here's an example of what a simulated set might look like. Here's what a real set might look like. And here's an early example of this. We're still getting all the regions and everything figured out, but how that can be used by the neural network, all that training data. And so interestingly here, my talk is more about training data and just thinking about how we're going to use all the spatial omics data. Then I'm not going into the details in the machine learning, but we can if there's more time. Human sciatic nerve is another really fun place to do this. Here's a normal nerve, just looking at the nuclei again. Here's a painful nerve, probably because of neuroma. There's this cluster of immune cells. We do something called SAHI and rotational ensembling when we inference, and that helps us really in complex tissue like this. These are not nuclei, by the way. These are axons that have really small sheaths wrapped around them in different parts of cerebral nerves and sciatic nerves from humans, actually. And so you can see that rotational inference and using SAHI, which helps you tile like massive images together, can get you really good data. And here's what one of those digital twins looks like. You can see these huge bundles of neurons, and this is not real. This entire image is all reconstructed from the machine learning, finding every little, axon, myelin sheath wrapping it, a Schwann cell nearby, perineural tissue, all kinds of stuff in there. And which again, we need to put those data sets together to do anything else. So I think I am pretty much at time. So I will zoom out. But yeah, anything you guys want to talk about? And if anybody wants to hear more about how we actually put the mutations in and screen the single cells, happy to talk about that as well. Thank you, guys.
Thanks a lot for the very nice presentation. So let's see if there are some questions from the audience. No. Okay, so I will start while they are warming up. I'm curious about the parameter that you use to generate your simulation, like the radius, for the cells, the distortion, if I remember correctly, yes, distortion for the section node. These parameters are from an available data set, or you generate them in the lab? How do you obtain these distributions?
We have some real tissue always, and then we get that image and we say, okay, here's a starting point. Here's the kind of, we can easily measure with standard techniques all of those, kind of all of those features. And so that gives us a really nice starting point. And then as we do that across multiple samples, we can build, here's a little atlas of the different ranges that we see. And, there's another thing that Daniel kind of brought up, the concept of, he wiggles the light, right? He makes these perturbations. We're also starting to think about how we can perturb even this is a node tree, right? This is like a differentiable tree of functions. And so using genetic or evolutionary algorithms, and I'll have to look up some of the techniques he mentioned. I think it was like SPM and SPSA, to actually have it help us get variation in a sensible way, and then use that when we're learning to see how those variations help us. We haven't done that in a formal way, but I think that would be super cool. But Matteo, I don't know if that's probably going off of topic.
Thanks.
So I might have an additional question, if I may.
Go ahead, Marco.
Yeah. So would it be possible to add, let's say, another level of complexity and somehow integrate the functional relationship between neurons or areas in the neurons in the pipeline, let's say? I'm thinking about functional connectivity, for example. Would it be possible to measure some parameter like this and integrate in the scheme?
So, I mean, I love that idea. Currently, we are not doing any like optogenetics or imaging where we can see these. These are everything except the bottom right, where I haven't told you about, is fixed tissue.
Yeah, of
And
course.
so we wouldn't be able to get, see how the neural activity is in these specific tissues. But we do work with groups that do that. Is that what you're thinking, where you actually have like activity data?
Well, no, I'm
going a different direction.
thinking about collecting functional readout, I mean, I don't know, in a slice, and then move to the fixed tissue later.
Yeah, I love it. We've thought about that a little bit, but yeah, we're just not exactly sure the right way to combine those data sets. But I'd love to get ideas from you.
Mm-hmm. Yeah, thank you.
Okay. I have one last curiosity.
Yes.
when have you tried in the section part, like some section in which you have like a tumor or something in which your anatomy there changed a little bit, how it's working, how it's performing in that situation? Or
Yeah, one thing I didn't mention is that instead of, so there's a lot of ways to do it. You can kind of get an example here, right? So in real tissue, there's tends to be the density of where we put these neurons in real tissue is fairly stereotypical, which is kind of what you're getting at. What if you get a situation where there's a tumor there, and so it really changes the stereotypical nature? This is really fun. So in our simulations, rather than us saying that the isocortex always has some range of densities, we were doing that early on, right? And then we were saying like, okay, fiber tracts have much lower density of nuclei, they have density of other things. And then like obviously the granular regions of the hippocampus have very high density. Rather than encoding that in per region, we instead switch to a system where we say any region can have any density. And that means that when we simulate, we don't force, so we're able to handle, for example, tumors, because it hasn't learned that this area always has to have density X and this area has density Y. It's learned that the difference in different regions and how they tend to connect together is what's important. And so if something pops up that that breaks that, it doesn't know what to do with it, but it still like understands that the other regions kind of can get pushed out of the way. What we haven't done is like put in random regions in between other regions, but that would also be fun to try to do. But yeah, having the densities be not locked into what reality always looks like has helped, because in a lot of really bad neurodevelopmental diseases, the densities do not look standard stereotypical. Yeah, great question.
I have a note for William. Sorry if it's unrelated, but I wrote a message in the chat. If you can.
Oh,
read
sorry.
it.
Go ahead. Yeah, I didn't see it yet.
Yeah. No, just read the message. It's
okay.
unrelated to this.
Got it.
Thank you.
Thanks, everybody.
Thanks.
Thanks to you. I guess it's time to move to the next speaker. And so the first and final presentation of today's webinar. It's a pleasure to introduce Professor Tommaso Dorigo, first researcher at the Italian National Institute for Nuclear Physics, member of the CMS experiment at CERN since 2001, and visiting professor at the Lund University of Technology in Sweden. Prior to joining CMS, he was a long-standing member of the CDF experiment at Fermilab, contributing to some of the most important development in high-energy physics over the past 3 decades. He also is based in Padua, where he's an art teacher and art professor at the University of Padua. And Professor Dorigo's research spans a broad spectrum of particle physics, including the studies of the Higgs boson, top quark, and as well as search for new physics beyond the standard model. And alongside his works in fundamental physics, he has made significant contribution to statistical methods for data analysis and inference, with particular interest in machine learning, anomaly detection, differentiable programming, and optimization of complex scientific measurements. So it's an honor to have you here, Professor Dorigo. Please, the stage is yours.
Yeah, thank you very much. I'm going to share my screen. Also, thank you for promoting me for professor. I've been a contract professor in at the statistical sciences department, but right now I can call myself a professor only because I'm a guest professor at the computer science department here in Luleå in Sweden, where I'm sitting right now. So let me share the slide. Yeah, but I'm a researcher, a first researcher at INFN Padova. That's more a better connotation of what I actually am. Anyways, I want to do the slideshow from the beginning here. Let's see if it works. Can you see the screen?
Yeah.
Yes? Okay, so this is an excerpt from a colloquium talk I gave recently at MIT. because much of the material is good for this talk as well, but the time is a half, so I will have to skip through a lot of material. I already removed some, but I still want to go through some of the stuff. And this slide deck will be available after post-mortem, so that in case you are interested, you can look at the stuff I actually had to skip. So we will talk about what I call the first ML revolution in fundamental science, and what I think the second revolution will be is when we get serious about rethinking the way that we design our experiment in fundamental science, and that is by doing the co-design of hardware and software for these experiments. And I will give one example I directly worked on recently. And then we will look at a few ideas that are there in this context for future applications to particle physics. That is the field of studies I am coming from. But also, we will also look a little bit into some neuromorphic computing applications to particle detector design. So yeah, I'm not going to discuss the context, particle physics and related experiments, the standard model made of building blocks of matter and interactions. That's what we study in these large experiments, the Large Hadron Collider, the CMS experiment that was mentioned a moment ago, and so forth. Big experiments, huge amount of thinking going into designing these experiments. And now we are digging the data that these devices are. Sorry for these long slides, and this is just a GIF that I will let run just to show how the data was collected to prove the existence of the Higgs boson. That is the main discovery that this machine has so far produced. And the Higgs boson signal is this red simulation peak at 125 GeV of reconstructed mass, which this is the data that proved the existence of that particle in 2012. And the fact that this happened in 2012 is not a coincidence. Well, it maybe is. I will not tell you how we got there, the way we extract this high-level information from these collisions, but the way we used to do this kind of business before 2012 was to look at single distributions of variables, and we didn't do multivariate analysis. Now, in 2012, this changed because deep learning was used for the first time for the Higgs boson discovery that you've just seen the relevant graph about. But the 2012 also marks the year when deep learning was used and became better than humans at classifying images. There was this large challenge, ImageNet. And so it was a paradigm change, right? And before, in particle physics, the field I'm in, nobody wanted you to use machine learning to publish data analysis. It would stand in a limbo forever in internal reviews. Collaborations would not want to publish. And after 2012, everybody is asking you, if you are not using machine learning, why aren't you using a machine learning tool, a deep neural network, to do the signal extraction? So really a paradigm change. And a further evidence that this was the case was a challenge that was put forth in 2014 when almost 2,000 teams tried to do this Gedanken experiment of separating this Higgs signal from backgrounds, all simulations, but this was just an exercise. And so it was proven, therefore, by looking at the winning solution in terms of the significance of the signal that could be extractable by using this DNN pooling that a computer scientist, in fact, did to win this challenge. You could get it was equivalent to having collected 6 times more data. That is a statistical power of square root of 6 in terms of of the discovery significance that you could get. So this means a lot of money, because just to run the LHC experiments every year, it takes a significant number of millions of dollars just in electricity, right? So this is very convincing. And there are signs of this revolution in the making if you also look at the papers, but I don't have to convince you about that, right? More convincing is really the change of mindset in the experimental physicists, my colleagues, that really now want to do this for the rest of their life. And in fact, we are doing it in many different directions. We are exploring higher energy machines, the collisions to find new particles, new forces of nature, many things. Also in astroparticle physics, neutrino physics. Also, theorists are using deep learning in many different ways. So that is what connotates a real revolution. And this is only software activities, but we are also detector builders. We build experiments, we design experiments. And so that's the next customer in line, really. The optimization of detector design, not just optimization of scientific inference from some data, but optimizing an experiment end to end. And that is what I call a second revolution in fundamental research, because when we will be doing this consistently, not only will we get more bang for the buck, every experiment will do the real best that they can with the allocated money and the resources, but really these AI tools will find a proper use in our field of studies. We are successful detector builders, but if you look at the way detectors are being constructed, these are cutaway views of a detector from the 90s and the CMS experiment 20 years later. And we are designing new experiments for the future collider experiments. And we are relying still on some paradigms that have lived with us, and they are worked very well. I won't go in the details of this because it's not the focus of this talk, but all these construction paradigms, which have served us well, don't allow us to go beyond and actually explore possibly better solutions. And that's also because the design space of these bigger, large projects has grown to transcend our understanding, so that it's very hard to explore this large dimensional space and imagine new ways to detect particles. These detectors are taking snapshots, pictures of the particle collisions and all the particles that fly through the detector. And they do this, for instance, at LHC every 25 nanoseconds. We take a snapshot with tens of millions of electronic channels. And of course, you could imagine many different ways to do this, but we risk doing the same thing all over again. And that's the danger, I think, right? We are not evolving. So who are you going to call? Of course, you're going to call AI. But the path to an assisted optimization of the design of experiments runs into hurdles. Of course, the high dimensionality of the design space, also the multi-target nature of the utility of these large experiments. And these processes that we are studying are highly stochastic. That means that every time a proton goes through your detector, it does a completely different thing, even if it has the same energy and characteristics. So we need to not only think about the hardware, but also the software extraction of this data, because sometimes you fix the hardware and then the software, thinking that you will do the inference extraction in some way. And when you think, when you crystallize the way that you're doing the inference extraction, then you cannot go back, and your optimality and the misalignment exists, and there's the interplay makes you not optimal. So all of this goes into thinking in terms of co-design. And if we think at the scale of optimization problems, it shouldn't scare us, because if we think at large language models, they now have billions of parameters or trillions of parameters. Um, I published a paper where I could optimize simultaneously 66 million parameters with a statistical learning tool. So the scale of a future detector for a future large collider is not even in that league. So, but yet it's not presently contemplated. Why we should do it? I get asked a lot, what do you think optimality is for such a large experiment that wants to make so many measurements and publish, you see, thousands of papers about different things? What does it mean to be optimal for a detector? You can see here on the right the two different experiments, the Atlas and CMS experiment at the CERN LHC, that both wanted very much to detect the Higgs boson, and so they needed to have very high resolution in the momentum measurement of their charged tracks. And here you see the invariant mass distribution of pairs of charged tracks, in fact muons, that come from the decay of three different resonances called upsilon states. And you can see that CMS has a twice better resolution in the momentum, so in the invariant mass of these particles. The peaks are better discernible over the background. So why does Atlas, which also spent even more money than CMS, got twice worse resolution? Because they decided to optimize their money differently. Then they decided they could live with a smaller, worse resolution in tracking to get a better resolution in the calorimeter. So, there are different ways to do this, but the problem remains, can we speak of an experiment-wide utility function? Because, okay, CMS can measure tracks well, but Atlas measure well something else. And where do you put your money? So multi-target optimization is pretty much a thing. And it's not so problematic. In fact, I spent some time to demonstrate that we are actually doing it in fundamental science. We construct multi-target optimization objectives and try to optimize them. So one clear example is one experiment in Beijing, which measures electron-positron collisions at various different energies and does a scan of the energy spectrum. And so in so doing can measure different particles and different phenomena. And in fact, there's a plethora of different measurements that this machine has produced and is producing. And they have to decide how much time to spend on each of these energy points. And they do it, and they have a plan. They say how many days they will run at the different energy points. And this is basically saying what the utility function of the full experiment is, because they do like to measure charmed baryon cross sections, for instance, but they also like to measure R values. And so all of these things are given a value. So we shouldn't shy away. It's possible to sit down in a table, call each other names, and then at the end of it, at the end of the day, we'll come up with a recipe for optimization. A single number, which can be the loss function of your machine learning tool. So what is co-design? Co-design is based on realizing that the achievement of a goal requires to tailor each part of a system to all others. So you can do holistic optimization and integrate the various elements in the best possible aligned way. Okay, and to prove that co-design is important, also in particle physics, is not immediate, but we spent some time and published a paper to prove that. So typically, you have a design space will be made of hardware parameters on the vertical axis and software parameters on the horizontal axis. You can think of this as different algorithms, different parameters of the algorithms, and so on. And if you fix the software reconstruction based on simulations that you are going to use to extract your information from some hardware, and then you simulate different possible layouts for your hardware, you can then study which of these performs the best. And note that hardware, that the simulation of this is very costly. You cannot populate this space with millions of points because these are stochastic processes, and a full simulation takes a lot of time. So you pick the best point given a software method. And then maybe you can retune your software algorithms and find the best point in the horizontal axis. But by then you are fixed the hardware. And so you are not likely to reach optimality in the true sense. This can only be achieved if you do a common optimization of all parameters by gradient descent or other tools, but gradient descent is very suitable. One example of this was a study I did of a very simple experiment meant to measure an ephemeral property of muon electron scattering. That was a contribution to a systematic uncertainty in a parameter called G minus 2 of the muon, which was a long-standing deviation from theoretical calculations and could be a hint of new physics. And this experiment just smashes muons on a beryllium target and measures the electron that gets kicked out along with the muon. And okay, I am a referee for this experiment at INFN, and I asked the proponents to come up with a optimize the layout, but they said that this was basically optimal, and they are detector experts. So I did the study myself. I simulated the processes, and I studied many things. Here you can see as a function of one parameter, which is the segmentation of the target into thin layers. Because why do you want to segment the beryllium target? Because if you do, you get access to the position where the interaction has taken place, because the interaction will not take place in air, but it will only take place in the denser beryllium. And this gives you a handle to measure these kinematics better. But if you don't do a vertex constraint in the reconstruction, so a software theme, you don't care about segmenting the layer. And that's what they did. But if you actually do a vertex constraint fit to the tree tracks, then you find that there is a highest utility region where you impedance match your native resolution from the track fits, the single track fits, to the spacing of the layers. So this actually provided an improvement in the relevant parameter for the optimization of this experiment by a factor of 2. So there you get a factor of 2 improvement in the output of the experiment, not spending more money by just thinking in terms of co-design. Okay, and this was published, and the collaboration actually took note of these suggestions. So by building a pipeline for end-to-end optimization, that means a software digital twin that can simulate the hardware and simulate the inference extraction, the pattern recognition, everything in between, and give you the final utility of the experiment, what you want to measure, and tell you how well you're doing it. Constructing this digital twin is very hard. And it requires the hand of computer scientists, analysis experts, and everything in between. But we are starting to do it. We have a collaboration that is growing fast and is precisely looking at these problems. And we have workshops and stuff. And there's also a community in Europe that's building around these concepts. We published a paper on co-design for scientific experiments, 90 pages paper, and we looked at many different use cases. Very quickly, because I know I have no time, but I will show you very quickly what an astroparticle physics experiment optimization looks like. This particular experiment looks at cosmic rays, photons that impinge in the atmosphere at very, very high energy from the cosmos. And by studying the direction and energy of these photons, we get to know about the most violent phenomena in the cosmos. And this experiment wants to build 6,000 water detector tanks that measure the Cherenkov light in that high altitude in Chile. And of course, if you have 6,000 units, you can lay them down in a cylindrical symmetry, because that makes sense in many different ways. And the collaboration was studying 13 of these possible layouts. Okay, I won't tell you why this is relevant, but you can imagine that very, very high energy gamma rays are important, and this experiment is looking at the very high energy part of this spectrum of electromagnetic radiation from the sky. Of course, Zwigo has to agree on the utility function. Even there, they have many different use cases for this detector. One might abstractly write the utility as a weighted sum of the flux precision, that is the flux that they get at a certain energy of these photons divided by the uncertainty on the flux. Okay, and when you have that, you can construct a pipeline. You simulate an initial layout for the detector, and you simulate the gamma rays, but also the background, which is constituted by protons. And then you reconstruct these particles so you see how well you're doing. And you can also try to distinguish the gammas from the protons and derive a test statistic that tells you how well you're doing this. And then it's lots of technicalities, but you need to have a PDF for this test statistic in order to understand how well you're doing. And you can do this by several techniques I won't describe here. And at the end of the day, you can actually write a likelihood function for the flux of gamma rays and use some statistical tools that tell you how well you're doing. And if you have done all of this in a differentiable way, you can get a derivative of the utility function as a function of the position of the detectors on the ground. And then you move the detectors on the ground following the gradient of that utility. And if you do this enough times, you will converge to the absolute maximum of the utility. So you see here you are both simulating the hardware and the software. And these are three different layouts. The black points are the detectors, and the colored points are the center of the showers that are simulated impinging on the layouts. And you can see that they get pushed around by the gradient of this utility. And if we run this simulation for long enough, you will see that they all converge to the same layout, signaling that indeed there is a highest utility point in this 600 dimensional space. This is constrained, not the 12,000 dimensional space that the full Zwigo experiment would require. But okay, so this thing works, and it basically takes into account both the hardware and the software in a combined utility. And so basically by coding up this required to me more than 1 year of my life and 20,000 lines of code. But I proved to myself that not only can this be done, but it should be done. Okay. So, okay, I think you can trust me if I tell you that these simulations converge. And so this is documented here. And okay, so this is the Pareto front of three different components of the utility function that we have just seen. And the design proposed by the Zwigo collaboration is laying here. And the third component is the color of these dots here on the plane. So after optimization, you see that you have moved the Pareto front significantly. So there is a lot to win in this kind of optimization tasks. So by getting a 30% improvement in your utility for an experiment that costs 60 million euros, it means that you have won 20 million to the experiment. So it's not idle, right? And I actually pushed this forward by showing some more co-design elements by having a very detailed cost model for the various components, fencing, cabling, leveling of the ground, the land cost, the cost of the electronics and everything. And you can code this up and you can actually say, okay, I have a certain budget here on the bottom right. I could actually use a laser pointer here. And you can say, okay, I have this amount of money. I don't want to spend more than this amount, but I don't even want to spend too little money. So there is a utility component that is a factor in the utility that has this kind of shape, and you can code it up. and add this to the other factors in the utility. And actually, this in this second load of the optimization of this experiment, I actually considered how the experiment would actually do the measurement of finding a 5 sigma signal of a photon source in the sky. And so the utility, the scientific utility in this case, is the inverse of the time that it takes you to integrate a signal that amounts to a 5 sigma discovery. And so the pipeline grew a little bit more complicated with reinforcement learning for the parts that are non-differentiable. And I want to cut the story short. Here is an animation again that shows the utility on the left growing as you are moving the detectors on the ground. And you can see the area of the Parque Astronomico in Chile where this thing would be built, and various other metrics that are improving as you go, and the cost model. So it's an example, but it shows that this can be done and that there are large factors of improvement that you can do. So you get more science out of the money that you get given for doing some experiment, okay? And indeed, the co-design part of this is that I could prove that if one would first optimize some part of the system and then proceed to optimize the rest, if one looks at the Pareto front, one sees that only the co-design that takes into account all parameters at the same time can investigate space of the parameters where actually the total utility is maximized. And you get to see that you have the full optimality is better than if you do things in 2 stages by a factor that goes from 6 to 20%. So co-design is really important in these endeavors because software and hardware parameters will talk to each other. I will not have time to show a similar example in calorimetry, unfortunately, but I will conclude because I think my time is over, discussing how we can extract and pre-process information from scintillation-based calorimeters, so the light that gets collected from complicated hadron interactions. This is a block of matter that is impinged by a proton. And recently, there is a lot of emphasis in doing particle identification, so being able to distinguish a proton from a kaon from a pion, because these tools have never been able to do that meaningfully. And we proved actually that this is possible if you have a very high granularity. So if you actually can reconstruct this pattern of energy deposition at very, very precisely. But this means constructing a 1,000,000 cell calorimeter that is very impractical. Instead, you can do this in a different way if you interpret these light signals and their time distribution using a neuromorphic computing processing. So here is just a slide that says that if you have a very small granularity, you should be able to disentangle protons from kaons or pions. And so there is a scale at which you lose that information because your calorimeter has too wide cells. But you want to know what is this scale before you build the calorimeter, and you want to know how much you can win in terms of discrimination power. We studied this. We showed that if you have very, very high granularity, you can actually distinguish particles. This is the first time it has been shown. Of course, you need graph neural networks for this. And so now what we want to do is to do the pre-processing of light signals from, say you have a particle coming in doing an interaction inside your scintillation detector, and then you have light that gets propagated back and forth and hits some sensors. These sensors could be nanowires. We are presently studying nanowires for this precise reason, because then arrays of nanowires can constitute what here is simulated by a spike in neural networks. And we have proven that this kind of tool that measures not only the amount of light, but also its time distribution can, and here you can see a movie of the kind of light that a grid of sensors would see from a particle interaction. And so you have a train of spikes that hit your spiking neural network or neuromorphic system that has a time distribution and an intensity distribution. And you can actually regress not only the energy, but also the position where this energy was deposited with a granularity that matches that of the very high granularity calorimeter we were thinking at the beginning earlier. I was showing that, and this is not granular, so you can bypass the granularity of the device by having a time information readout. And so this opens new ways to detect and measure these particles. And in fact, we are investigating these nanowires and arrays of nanowires that do the computing on chip. And this is actually the subject of a winning Pathfinder project that we have started in April with colleagues in LTU, NanoLund, and other. It's a consortium that includes Hewlett-Packard Enterprise. And what we want to demonstrate is that you can go beyond by using neuromorphic computing for the preprocessing of these light signals. And okay, there are also longer-term plans for the hybridization of tracking and calorimetry, but I don't think you're interested in this. So I will just look a little bit into the future. We have a Large Hadron Collider. It's this small thing here in this aerial view that imagines a future circular collider, which is actually the main direction CERN has taken for the future. So if we build this 100-kilometer tunnel under Geneva, it will take 20 years to build. And the Chinese also want to do something similar. But if we do this in 20 years, imagine what AI tools we will have available in 20 years at the pace we are going now. So we should not only seriously consider co-design optimization of all the detectors, but also we need to think to make a leap forward and think at what AI tools will be available then. So I think the key to doing this kind of exercises to design experiments having in mind a much better inference extraction that is possible with the tools we have today. So, okay, this is stuff for the future, but in general, we need to move away from experience-driven and robustness ensuring ideas that have governed the design of particle detectors for the last 60, 70 years, because they actually constrain us and prevent us from discovering new avenues. And I've given you some ideas of what could be these new avenues. Actually, we didn't have time to go into this possible hybridization of tracking and calorimetry, but we need to seriously yield power to our AI helpers in these problems because humans cannot compete any longer. So I thank you for your attention, and I'm happy to take your questions.
Well, thanks a lot. At this point, Professor Tomás Rodrigo, the promotion is for free. Well, let's see if we have some question from the audience. Well, as an outsider, complete outsider about this subject, I might have a very low-level question.
I'll take it.
Yeah, okay. This is for free as well. And that would sound something like, would it be useful to, I don't know, use something like generative model to develop hypotheses instead of the optimization of detection on all the stuff that you presented here?
Yeah, so we are indeed doing that. We are using generative AI because we need to bypass one important hurdle in the optimization, which is the stochasticity of the physics processes. So, we have a tool that's called GENT4, which is a full simulation of these physics processes and the interaction of particles with matter. And it takes a long time to run these simulations. They are very, very precise. They are high level, high fidelity simulations, but we don't need them high fidelity for optimization tasks. Rather, we need them to be fast and we need them to be differentiable. Because if we cannot pass gradients through this pipeline, we cannot do stochastic gradient descent. And in high dimensional spaces, other optimization techniques don't work nearly as well. So what we need to do is to create surrogate models of these stochastic processes. And we do this with generative AI. We do it with the diffusion models typically. We do it with transformers. We have been using these tools for a few years now. There's a significant amount of knowledge that has been developed to do this. Surrogate modeling is really at the heart of all of these tasks, not only in high energy physics, but also astroparticle and not just in detector design, but also just in the software analysis, because these simulations are really very costly and they are the bottleneck. And by doing generative AI, with generative AI, we can bypass this.
Yes, thank you. Perhaps another question on this line. Would the use of the adversarial network be useful in this way? I mean, you might train adversarial network based on the results on the simulation and the optimization of the detector phase, for example.
Yeah. So this is indeed one of the tools that is used when you want to verify that you have a good enough surrogate. You can test it against the full simulation, or you can by adversarial techniques, or you can build a generative tool that learns the structure of your data by adversarial methods. So I must say that these GANs are used in particle physics more in the software than in these particular areas that I have been discussing, where diffusion models and transformers and normalizing flows are preferred as ways to obtain decent level simulations of multidimensional features. But they pertain to the arsenal of tools that we employ for this task, for sure.
Thank you. Any other question? Perhaps from my colleagues?
I actually have a question. I'm not an expert either in this field, but I believe it's very interesting that we kind of see some bridging between techniques developed also